Good evening, students. This one is the chapter of biotechnology and its application. In this chapter, biotechnology, you have to find out what are the uses of a biotechnology and its product. So, not spending so much of time, previously you all understood about each and every chapter. So, this chapter is in the different way. Here, the biotechnology actually a type of a field where any type of the DNA is isolated, जहाँ पर से कोई भी DNA isolate किया जाता है and then it is going to insert inside a vector and make the different number of the copies. The DNA is choose according to the gene of interest and it is inserted inside the organism to change the phenotype of a particular DNA, and this method which is used here is used in the past few years. There are many applications where this type of biotechnology are used in the field. The fields are agriculture, energy and environment, healthcare and pharmaceuticals, diagnostic, food innovation, and fermentation. These three research areas of biotechnology is due to improving any organism which act as a best catalyst, which used as a catalyst is going to improve by the help of biotechnology to create an optimum condition for the growth and action of the microbes. And the third one you already know, downstream technology. So where this application is going to apply, the main areas where these applications are going to be applied. First of all, why it is applied? It is applied to increase the quality and quantity of the food crops. And it is achieved by some methods. The number one is agrochemical based agriculture. Number second is organic agriculture. And number third is genetically engineered crop, which is the best part. So in agro-based chemical agriculture, actually it is creating a pollution. And because of that, this is going to be rejected. Organic agriculture is expensive for the farmers. Mostly the farmer are not able to spend the penny on this. That's why organic agriculture is different. And the third one is genetically engineered crop based agriculture, which is actually the main motto of this chapter. Because of that, I just want to say that biotechnology is the integration of a natural science and organism cells and parts so that the different type of the product and services can be obtained. Any genetically modified animals or any genetically modified plant or any genetically modified medicine is formed by the help of biotechnology. Now the green revolution actually took place in the last 60s. Norman Borgman, which is also called as the father of a green revolution, tried to increase the agricultural productivity in the world, but can't be possible at that time. In India, MS Swadminathan also gives some hands to increase this revolution and encourage modern methods because these methods are due to increase the yield of a crops done by providing the improved variety of a seeds and also improving the irrigation facility with good use of agrochemicals. But for the farmers of a developing country, it's very difficult to afford these agrochemicals, as I already told you. So the only way which we are going to use is genetically modified organism. The best answer of this question is the field of biotechnology, which can be obtained easily by the help of different plants, animals, and microbes. Genetically modified plants and crops are very useful. They are being used by a farmer, and why they are using, because of some advantages. But before that, we are going to find out what is genetically modified organism. It is any microbes, any type of the fungi or any animal, those genes are used to produce the genetically modified organism. Like as a golden rice. Golden rice is actually a type of a rice which has a high quantity of beta carotene, which convert any type of the provitamin A, which beta carotene convert vitamin A into metabolized by the help of human, and because of that the color of the rice become yellow. That's why it is called as a golden rice. So we back to the part, what are the advantage of genetically modified plants? So there are some. First of all, they are tolerate to abiotic stress. Lots of stress are present in front of the farmers, and these farmers are can't able to tolerate with these type of the abiotic stresses. The second one is the pest resistance crop, which is reducing the harmful chemical. By the help of these pest resistance crop, harmful chemicals are ignored. It also reduces the post harvested loss. The previous loss is like as a when you grow any one single crop, the single crop use all the type of the humus and other nutrition of a soil, and because of that the next crop, before sowing that, we have to think about something, that how can we obtain the humus again in this soil. So they also reduce the post harvest loss. They enhance the quality, like as a golden rice, you already say vitamin A enriched, and they also increase the ability of a mineral uses by the plants. So the production of a pest resistance plant is very important. The first one is Bacillus thuringiensis, which is also called as a BT, a type of a gene which actually has a property of a pesticides. The genes are obtained by the bacteria and from the different parts of the plant, and produce them a resistance against the pest. Actually, it produces a protein called as a cry protein, and that protein is responsible for the insecticidal activities. This protein kills the lepidopterans like as a tobacco budworms, coleopterans like as beetles, and dapterans like as flies and mosquito. This bacteria, the bacteria Bacillus thuringiensis, produce a protein crystals which contain an insecticidal protein. The crystals does not kill the bacteria, but this Bt toxin protein is in the inactive protoxin inside the bacteria, and the pH is alkaline, which is required to activate the crystal. When it goes inside the body of an insect, or you have to say when it is eaten by the insect, the alkaline pH of a gut, the gut of an insect consists of alkaline pH, make the inactive protoxin into a toxic form, and this toxic bind to the gut epithelial lining and create a pores, which causes the cell to become swell, and then it become broken down, and because of that the insect become killed. The toxic synthesis consists of 4 or 5 types of a protein of a cry. Like as example, these cry protein are in the different type of the work on different type of organism, like cry 1AC and cry 2AB. It controls the cotton bollworm. Cry 1AB controls the cotton borer, controls corn borer. Cry 3AB control the potato beetles and Colorado, and cry 3BB control the corn rootworm. So basically different type of a cry protein and gene is responsible for the different type of pests. Now after that we can say there are many type of the insect species which are sensitive against the cry protein, and due to their high affinity of these protein, such insect species seems to be cleaved with several fragments, and because of that we stop the growth of pesticides, insecticides. So the next one is RNAi, which is called as a RNA interference. What is that RNA interference? So in this, the root of a tobacco plant is infected by a nematoda, which is called as Meliodogni incognita. It affects the yield of a leaves, and this strategy is developed by the help of the RNAi. I will have to show you by the help of a flowchart. First of all, this nematoda consists of a specific gene which is introduced inside the host, and host is the tobacco plant, by the use of a vector which is called as Agrobacterium tumefaciens. So this AGBR act as a vector, and the production of a and it produce a sense and antisense RNA in a host cell. Two RNA become complementary to each other, bind together and form double stranded RNA. Why this RNA is going to be formed? Because by the help of this RNA we have to silence the gene. Now what is going to be happen? The RNA interference is going to initiate, and this double stranded RNA is cut into 21 to 23 segments. These segment is called as siRNA. These siRNA are cut by the help of enzyme dicer, RNA dicer. Now, these small fragments are going to use by the help of ribonuclease combined and form RNA inducing silencing complex. And this silencing complex cut the mRNA, cleaves the mRNA. If the mRNA is not formed, the mRNA of Meliodogni incognita actually infect the tobacco plant. Now it is going to be broken down by the help of RNA interference, that is RNAi, RNA inducing silence complex, and because of that it not going to produce a protein, and the growth of a tobacco plant is easily happen. I will show you one diagram. This is the double stranded RNA. By the help of this dicer, we cut the RNA and form siRNA. Then nucleotide is going to combine with it, and because of that the double stranded becomes unwind, and it cleaves the mRNA into smaller parts. This is called as gene silencing. Now I think you can easily understood by this flowchart. What are the application of this biotechnology in medicine? The application of biotechnology in medicine is going to produce genetically engineered insulin. This is the next part. Going to produce a gene therapy, going to produce a vaccine, and doing the molecular diagnosis. So, what are transposons? Sometimes the question arises. These are the units of DNA which can move from one DNA molecules to other. So basically, very important thing. Now we have to find out one by one. This genetically engineered insulin is actually a type of insulin which consists of three chains: the A chain, the B chain, and the C chain. Actually, many of the peoples in the world are suffering from diabetes and they need a treatment. The treatment is only insulin. Previously, in the previous part, insulin is isolated from cattle and pigs, which gives and suffering the people, those who don't have a insulin or have a insulin deficiency. It causing a lot of problem. So a large number of cattle are going to killed for this patient. Now it being creating side effects. This is the only matter that what are the alternative techniques, how can we find out this insulin. So human insulin is actually looks like as a dimer, a one chain which consists of 21 amino acid called as a A chain, another 30 amino acid called as a B chain which linked by a disulfide bridges. In mammals, insulin is synthesized as prohormone which contain a extra stretch called as a C-peptide in mammals only, animals. This C-peptide is removed from prohormone and then the insulin become mature. The main challenge is to assemble these two chain of insulin by rDNA technology, recombinant DNA. So here you see the B chain, C chain and A chain. The simultaneous folding creates a problem and this C chain is going to removed. After that, the A and B going to form a disulfide bridge and then it become a mature insulin. The first company who made this insulin in 1983 is called as Eli Lilly. And this DNA sequence of 2A and B is introduced them by the help of PBR322 of E. coli, a plasmid. Chain A and B are produced separately and joined by a disulfide bond. The second one is gene therapy. So what is gene therapy? Gene therapy is actually a set of ways in which any gene is defected in embryo or child can be diagnosed and corrected. First clinical gene therapy is given in 1990 to a 4-year-old child which is suffering from ADA deficiency, adenosine deaminase. This type of a disorder is due to delete of a gene for adenosine deaminase. This disorder sometimes cured by the bone marrow transplant, but it can be treated with enzyme replacement therapy in which ADA enzyme is injected into the patient. In gene therapy, the normal ADA gene copy is produced by cloning, and these genes were packed into a defective retrovirus. Now, lymphocytes were isolated from the patient, and this patient gives the lymphocyte with the help of retrovirus, which create a functional ADA, and the cDNA is going to introduced. These modified lymphocytes were injected back inside the patient. So here we are going to cure the lymphocytes. This method is not a permanent cure because the lymphocytes are going to be died after a few days. But new genetically modified lymphocytes need to be inserted again into the patient. So permanent cure is only the bone marrow transplantation. So here the diagram is going to say the patient which has ADA, the lymphocytes are going to be taken. Inside the lab, the culture of lymphocytes where the functional gene is going to transfer inside the lymphocytes. And then these lymphocytes are again cultured and make a copy, and the copy of these lymphocytes are again infused inside the patient. So this is a type of a gene therapy, a type of a collection of methods which allow the correction of a gene defect that can be detected in the embryo or a child. The next one is vaccine production. Vaccine is very important and produced by rDNA technology because there are few methods. First one is generating the antigens. Antigens are generated from the microbes. These antigens are the part of a disease causing microbes or a part of a protein which weakened or inactive form. Number second, isolation of antigen. These antigen are isolated from the culture and then they are generated. Then adding the preservative and adjuvant, these pesticides are added to the vaccine and multi doses are used while the adjuvant increase the immune response of the vaccine, which increase the storage life. And the fourth one is molecular diagnosis. Very important to detect the pathogen in the body in early days is really a best method to cure and possible. But traditional ways in the serum, blood or urine analysis, it is very difficult to detect in early stage. There are some early detection of the infection which can be possibly used like as a recombinant DNA technology, polymerase chain reaction and enzyme linked immunosorbent assay called as an ELISA. Recombinant DNA technology is actually the cells are going to clone from the patient. The probes are used in a single standard DNA, and then you already seen these things in the previous chapter how we do the recombinant DNA technology. The polymerase chain reaction is actually the presence of a pathogen which can be detected becomes given a high number of a body individual, but by that time the infection would have spread a lot in the body. So PCR methods are going for the amplification process. In the previous chapter I already told you about the amplification process. And this ELISA is actually a type of a technique by which AIDS are going to be cured. It detect the actually AIDS are not cured. AIDS we can detect the AIDS by the help of ELISA. It based on the principle of antigen antibody interaction. Small amount of the protein by the help of enzyme. Protein like as a glycoprotein or detection the antibody synthesized against protein. Now there is a question arises also sometimes what are stem cells? Stem cell are actually undifferentiated cells that are able to develop into a type of a tissue and perform a specialized function. Stem cells obtained by the help of bone marrow, umbilical cord, inner cellular mass, and embryo or amniotic fluid present inside the woman's womb. The next part is transgenic animal. Those animals whose DNA are modified and express the foreign DNA is known as transgenic animal. It may be rabbits, rats, pigs, sheep, cow, and fish. Animal those whose genes are being modified and expressed is called as a transgenic animal. The steps to form a transgenic animal is firstly we have to identify and then isolate the gene of interest. Then select a vector, insert the desirable gene into a vector by the help of enzyme ligase, introduce the vector into a cell, tissue or embryo, and then express the foreign gene in that transgenic animal. So the modification is used to done a normal physiology and development, like transgenic animal is used to study normal working of a gene, also study the effect of insulin growth factors or the genes from other species which are introduced and what are the effect of that. Now it also used to study the disease. Transgenic animal help to study the disease and we can find out the cure of the disease. They also give us the model of human disease. Now a time transgenic animals are used to cure cancer also, cystic fibrosis also, Alzheimer disease also, arthritis also. So before going to the further part, the reason of a transgenic is to study the disease, vaccine safety, defective organs, normal physiology, biological product and chemical safety. The first transgenic plant is tobacco plant and the first transgenic animal is the mice which consist of green fluorescent protein. Now the next part is biological product. The biological product is transgenic animal has a like as a human protein like as antitrypsin which treat the emphysema, and also there are some attempts made to the treatment of phenylketonuria and cystic fibrosis. In 1997, first transgenic cow Rosie is produced, whose milk has 2.4 gram per liter of protein. The milk contains the human alpha lactalbumin and it is nutritionally very a balanced product of human babies than natural cow milk. The vaccine safety actually the animals are being developed for the use of testing. Like as a transgenic mice are used to test the safety of a polio vaccine. If the mice are safe, then the years will come will replace the use of a monkey. It is done with the stepwise method, like firstly going to the smaller, then larger, and then in the human. The ethical use is the modification of living organism must be monitored. The animals need to be regulated and checked by the help of the product which is fit for the uses. In genetically engineering approval committee that make the decision whether this vaccine is going for the further use or not. Sometimes the genetically modified crop have consist of some controversies in turmeric, neem and basmati rice. Example: India faced a patent issue related to neem, turmeric and basmati. Like US Patent of Office, that is University of Medical Center of Mississippi, used turmeric to heal the wound. R.A. Maleskar, an Indian scientist, challenged the patent. The patent is a type of a technique by which we can prove that this product is only published and introduced or subjected to be discovered by this country. The basmati rice also offers some type of a controversy that US say that it is the patent and the trademark of a basmati rice is from their country, but actually the patent is prevented by the people who selling the basmati rice. Later patent are going to be revoked and it comes under the section of a biopiracy. Now what is biopiracy? Biopiracy actually the use of a bioresources by multinational companies without the proper authorization. If the authorization is not good, if the authorization is not correct, then you are not able to stamp the name of your country that this product is patent from your country. The Indian Parliament has recently cleared the second amendment of Indian Patent Bill and takes such issues into consideration and try to prevent such type of exploitation. So, biopiracy is the use of bioresources by multinational companies or other organization without proper authorization. Biological warfare is the use of an infectious agent like bacteria, fungi, and virus insect to harm or kill the humans or animals. That is also creating a type of a biowar. So the key points are there. You have to see it. Now, the biowar is actually also a type of a thing by the use of these biological living product. We are going to intermix in water, in food, or in air to kill the other countries' people. Like you say, coronavirus is also a type of a biowar, come under the category of only this. So, here there are the some actual photographs of flowchart by which you can understood about the previous what I told you and learn it by the depth of your heart. You learned it, make some intellectual questions and perform the answers according to your beneficiaries. So it is very important to understood these things in such a case. Thank you. Have a nice day, and you have to like, share, and subscribe. And also one more thing: go through the video, go through the chapter, and listen each and every word what I am saying very clearly. If you just only see this, you can't able to understand by yourself. So it is very important to listen the words very clearly. Thank you. Have a nice day. And tomorrow I'll also publish one of the chapter. So be good. Thank you.